Hi everyone. In this video I wanted to talk about the TLC of dyes experiment. So what you're going to end up with is some example ransom notes, and they're not going to look exactly like this, but mine is just test note that I made with my blue pen. I used the Bic pen, and then down below you're going to use a different sheet of paper than your ransom note is on, but down below, so I didn't have to waste paper, I just made a example of this blue pen. So basically what I did was I just scribbled in here until it was mostly blue and not that much white. So that was basically what I'm doing. Now what I want to do is use TLC to attempt to figure out whether this test note writing is the same as this blue pen. Now in this case I know it is because I drew both with the same blue pen, but in your case you're not necessarily going to know that. So this is what you're going to do. Now I do want to point out that the TLC would not be a confirmatory test here. It would maybe be a preliminary test. But even if you had, some kind of sophisticated mass spec on these molecules, it might not necessarily be a confirmatory test, because my guess is that the ink in a blue pen from Bic is possibly not proprietary to Bic pens, and it's definitely not proprietary to this blue Bic pen. The other blue Bic pen I have sitting over there probably has the same ink in it. Right? And if we got another blue Bic pen in another part of the country or the world, it would probably have the exact same ink in it as well. So, I'm not positive that this is particularly, damning information against a suspect. But at the same time, it is another piece of the puzzle that may give some kind of idea of whether or not the ink came from the same pen that maybe was found in the suspect's, place of residence or whatever the case may be. So that's basically that. So I want to talk about the practical aspects. You can see here I have my little tube, my microcentrifuge tube. I have my example ransom note and my example blue pen. But I do want to talk a little bit about the theory before we do that. So basically, we're looking at different types of dyes and pigments that we could find in ink. And here we have two anthraquinone dyes. All right, we have dispersed red 60 and dispersed blue 60. And some of these anthraquinone dyes are water soluble. Specifically, some of them have sulfonic acid groups, which is basically an SO3 minus deprotonated sulfonic acid group on them, that can make them to be water soluble. These two particular ones don't look particularly water soluble to me, but these are different dyes that you could find. What is something to notice in common about these things? Well, they have these six-membered rings, these three six-membered rings in common, okay, which is what makes these anthraquinone dyes. And also what they have in common is a large conjugated pi system, which means they have double, single, double, single, double, single bonds all throughout them. This makes these molecules able to absorb visible light, and molecules which can absorb visible light tend to have a color, and then that makes them perfectly useful as dyes. So for example, this one appears red, so therefore it would probably absorb green light, okay, which is somewhere around 520 nanometers if we were going to put it into a spectrometer, okay, because they absorb their complementary color. Said another way, white light is hitting a dye, and when you take away the green light from the white light, what you have left is red. So this would absorb a color of visible light, and then it would appear a different color. Another type of dyes that we have is phthalocene, phthalocyanine dyes here, which look like this type of molecule here. Now in this case, you can see that this has these two protons in it, but this is also known to be stable, losing those two protons and having a 2- charge, which makes it ionic, which could make it coordinate to a metal, or it could also make it potentially water soluble, depending on the case. So this is another type of dye. Again, you'll notice it has this pi conjugation, okay, throughout it, where you have this double, single, double bond type situation. And the last thing we have here is an Azo Pigment, which has this azo group, which is this N double bond N. Again, it has the same single double bond conjugated character. These molecules tend to be not water soluble. They also tend to be a little bit longer lasting and a little bit more resistant to light and things like that. And a pen could have any combination of these things. So if you take, for example, a black pen, well, what is black? Black is the absence of color. So we need something that absorbs all the colors. Well, finding a single dye that absorbs all the colors of visible light is probably unlikely. So it would probably be a mixture. And as you know from your previous experience with TLC, TLC can be effective at separating mixtures. So basically, if we were to spot one of these dyes on a TLC plate, or a black pen, the ink from a black pen, for example, then basically we could elute that through TLC, separate it into its component colors, and then get an idea of what component pigments that pen is made from, and then compare it to some unknown. Again, is it completely confirmatory that someone's innocent or guilty? No. Could it be part of a larger picture of evidence? Yeah, it certainly could be. So that's basically what we're attempting here. So what we're going to do is we're going to extract these dyes, and I'll show you how to do that in a minute, and using pyridine. And it's very important that we're only using 1 or 2 drops of pyridine. And it's also very important that the pyridine picks up the color pigmentation. So if the pyridine doesn't appear to be roughly the same color as the original ink, it's going to be a good idea to add more ink just to make sure that it's colored. Then you're going to spot it on the TLC plate a few times. And the TLC plates, you're going to have an unknown, that's the ransom note. You're going to have the suspect's pen, which is the pen found at the suspect's house. And then you're going to have 4 standard pens. Now, if your ransom note is black, I would hope that you would pick 4 black pens to test as your standard pens. If it's blue, pick 3 blue pens, so on and so forth. But use your best guess, to guess which one it could be based on the color of the ink. If there's any other things about the ink that make you think that the ink came from that pen, use those as well. So if it puts down a specific pattern or thickness of the line or whatever the case may be, yeah, certainly use those things to help you. So the first thing we have to do is we have to collect these ink samples. And it's not quite as straightforward as just taking the ink from the pen and putting it on the TLC plate. Now that could work theoretically. You could just literally write a dot on your TLC plate. The problem is 2 problems. One, you don't have the original pen when it comes to the ransom note. So this is my test ransom note. And secondly, you're probably going to put way too much ink on there and you're going to end up with a smear. So instead of that, what we suggest you do is for each of your standards, whether this is the suspect's pen found at their location or one of the pens that you suspect may have been used, make a box like this. Now, does it need to be this exact size? No, but make some kind of scribble. It could be a circle, or it could be a box, it could be whatever you want, but make some kind of scribble like this. Then what you're going to do is take your hole punch and make sure that there's nothing in it. Okay, so I'm just making sure there's no hole punches here. So this is kind of your hole punch collector. All right, this isn't usually used for forensic application. This is to minimize the mess when you're punching holes in things. So that's basically that. So make sure it's empty. So there's no cross-contamination here. Then what you're going to want to do is take this and put it over your sample like so, and then systematically, if you look off to the side, you can see exactly where the hole is going to punch, and then systematically punch holes in your samples like this. All right, so I made a layer of holes like that. Now I can go just next to that and make another line of punched holes. And I want to get somewhere around 20 to 30 of these punched holes. And for the standards, 20 or 30 punched holes should be plenty because there's tons of ink on here. Way more ink than what you're going to find in your sample where you actually have some writing. So once you have that, you can go ahead and open this up, kind of knock it out, and then what you're going to want to do is put these things into your vial. All right, and you want to get 20 or 30 of them, and you want to just kind of tap it so you can get them all down in the bottom of your microcentrifuge tube, kind of like that. All right, and I didn't do all 20 or 30, but you could see, maybe you can see, there are some in there. Okay, so now I have some in there. Now what I'm going to do is I'm going to add 1 or 2 drops of pyridine. I'm going to close this. I'm going to gently shake it around, and I'm going to let it sit for 10 minutes or 15 minutes. What I'm checking is that my pyridine becomes blue. Okay, if the pyridine solvent, I'm not going to do this because I'm in my office, I'm not in the hood. You're going to add the pyridine in the hood. I'm going to check to make sure the pyridine turns the color of the ink, or at least a color. It may not be the exact same color. It might be a lighter shade of blue or something. But I'm going to make sure that the ink is dissolved into the pyridine. Once the pyridine is there, only 1 to 2 drops. More is not better, because if it's too dilute, you're not going to be able to actually see your sample. So after you have your 1 to 2 drops of pyridine in there, and you've let it sit for a few minutes and it's turned blue, then you could spot it on the TLC plate a couple of times. It's probably a good idea to spot it on the TLC plate, let it dry, spot it again, let it dry, maybe even spot it a third time, let it dry. So you can see some kind of faint blue color on that TLC plate. And that's basically what you're going to do. And again, you're going to use 20 or 30 of those little dots. Now, to do your sample, the first thing you want to do is double check your hole punch catcher thing here and make sure there's no cross contamination, so there's no little dots here. And I'm going to close that off. Then what I'm going to do is I'm going to try to get my sample, and you can see here I spilled a bunch of my dots, so I could just make sure that those end up in the vial. So what I want to do is try to get this sample. Again, it can help to have this so that the punch part is on the top, and you basically look at it from the side and you can see exactly where it is, although that camera view is pretty nice, and try to get it over the letters as best you can. So you're punching out as much of the actual ink as possible. And you can just repeat for as much as you feel like you need. Now it says 20 to 30 dots. You may need a little more here, because if you look at the dots, they're actually not going to be completely colored because the hole punch is thicker than the letters, at least in my case. You also might need a pair of scissors. So this will only go, let me show you. This will only go so far into the paper, and I can't get any further. If you get to a point where you can't reach any further, you can use your scissors, just cut the paper down a little bit. All right, it does ask you to photograph the evidence before you start to destroy it in this way. So again, get yourself 20 to 30, I'd stick more towards the 30 side, dots of this ink. Then what you're going to do is put them in a different centrifuge vial, add 1 to 2 drops of pyridine, let it sit for 10 minutes, shake gently. Make sure that you see a color in the pyridine, and you're going to spot those on the TLC plate. So you're going to prepare several samples. Again, the samples that you are going to prepare here are the unknown, which is the ransom note, the pen from the suspect's house, and 4 standard pens that are the same color as, or have some other characteristic that leads you to believe that they are possibly from the ransom note, and you're going to spot each of those. So you're going to basically have 6 little microcentrifuge tubes with these dots in them, and with 1 to 2 drops of pyridine in each one, in order to extract off your solvent, or excuse me, your ink. You're then going to spot your ink on the TLC plate. You're then going to develop the TLC plate exactly the same as you did last time. This time you're going to make your eluting solvent with 14 milliliters of ethyl acetate, 7 milliliters of ethanol, and 6 milliliters of water. This will in total give you 27, I believe, milliliters of eluting solvent. This may be too much, so if you don't need to use all of it, just pour a little bit on the bottom and make sure that the eluting solvent is below the level of the TLC plate. Also, in the picture, we're only developing one TLC plate. You could certainly put another one in the other direction and develop both TLC plates at the same time. So you're going to go ahead and elute the solvents. Then what you're going to do is work up your results. So take a look at the TLC plates, see if you could see different inks move different directions. It would be a good idea to visualize them under the UV light in case there's something there that is faint and you could see it better under the UV light. However, these are colored dyes, so you probably will see at least something with just visible light because they do absorb visible light. They probably also absorb UV light, many of them, so it's not a bad idea to put the UV light over there and just see if you see anything extra. So that's basically what we're going to do. Then based on what's going on, make a guess as to which pen that ink on the ransom note came from. Said another way, which spots from your unknown, from the ransom note, look similar to the spots from either the suspect's pen or one of the standard pens, and then make your best guess. Again, this can't be really confirmatory because there's so many Bic pens in the world. It's very hard to say that it was this exact Bic pen. I would say it's impossible to say that. So therefore, again, it could be just another piece of evidence. I do want to remind you that it is very important to take a look at the introduction and conclusion sections. I should have mentioned this before I got started, but we covered many of the things in the introduction. Okay, so how we're going to find the ink used in the evidence, how TLC can be used to conclusively determine whether this came from a specific pen. We talked about how it can't and why it can't, and a general explanation of some of the general things you could find in ink. We talked about the three types of dyes and pigments that you can find in ink and how many inks are a combination of these different things. So I think that we left a little bit of the theory out today on this video because we spent a lot of time on the theory of TLC on the previous video. So here is just a forensic application of TLC that kind of coincides with what you learned about TLC last time. So thanks for watching, and I hope you found this video helpful.